These arterioles will then divide to form the capillaries. These capillaries form a network within the tissue known as a capillary bed. If you'll recall, we mentioned that the capillaries are the smallest of all the blood vessels, and as such, they have the narrowest lumen. The diameter of the capillary lumen is about 7 micrometers, which is about the same as the diameter of a red blood cell. So these capillaries are just wide enough to allow the red blood cells to squeeze through them. This small size of the capillaries allows them to bring the blood as close to the cells within the tissue as possible. Now, if you look closely at the single layer of endothelium that lines these capillaries, you'll see that these cells are not exactly packed tightly together. In fact, most capillaries have tiny gaps between the cells in their walls, which allows plasma from the blood to seep out and leak into the spaces between the cells in the tissue. These leaked components of the blood fill these spaces and help form what is called the tissue fluid. This fluid bathes all of the cells in our tissue, and it's through this fluid that substances will be exchanged between the cells and the blood. Now, by the time the blood gets to the capillaries, it's at a much lower pressure than when it first left the heart. See, blood in the aorta is at a pressure of about 120 millimeters of mercury, but by the time it enters the capillaries from the arterioles, it's at a pressure of about 35 millimeters of mercury. Thanks to this lower pressure and the narrow lumen of the capillaries, the rate at which blood is flowing through the capillaries is much slower. This slower flow rate allows enough time for the exchange of substances to occur between the blood and the cells in the tissue. Remember, it's the capillary bed which is the main location where exchange of substances between the blood and the cells will occur, so it's really important that we have enough time for this to happen. By the time the blood gets across the capillary bed and reaches the other side, it's now at a pressure of around 10 millimeters of mercury. From here, as these capillaries leave the capillary bed, they will start to join together to form the venules.